I don't have too many slides, so hopefully that leaves plenty of time at the end for questions. But I'll kind of give some background on some of the work we've been doing and kind of building off, um, uh, moving from some of our published work into some new projects being spearheaded by new students in the lab. So I'm always told to start with a slide where everyone knows and is familiar with. So here's that slide. So during physiological erythropoiesis, red blood cells are produced by a cascade of pluripotent and multipotent cells, leading to efficient red blood cell production at very high rates during physiological processes. This rate has to increase in contexts of regenerative erythropoiesis, for example, acute and chronic anemia contexts, where either not enough red blood cells are being produced or the half-life of mature red blood cells has been decreased. And we know from work by Bob Paulson's group primarily, but also Merav Sokolovsky and others, that there is a separate process that is involved in regenerative erythropoiesis, in which erythroid stress progenitors get activated through a variety of signaling cascades to contribute to the replenishment of red blood cells that have been lost and to help restore homeostasis. And so one of the kind of central features of the work that we do in our lab is trying to identify the unique mechanisms that are involved in driving the progenitor function during the context of stress and acute anemia. And that, if we can better understand what the genetic features are that are defining this phenomenon and the proteins that are involved in it, then we can identify new therapeutics and diagnostic markers for treating hematologic disease. So I'll present two projects related to that today. The first project is from a former graduate student in the lab, Yi Chao. I think I presented very preliminary data on this the last time I presented to this group, maybe 2021 or 2022, kind of trying to develop a comprehensive map of transcriptional changes occurring in acute anemia and linking those to specific genes and proteins that are up and down regulated in the context of acute anemia. And then the second project I'll mention today is on kind of predictive models to identify the upstream requirements for transcriptional changes that are occurring in acute anemia. So these projects all began kind of with our work on a protein called SAMD14 and its cis-regulatory, intronic cis-regulatory element that controls its expression in anemia. This EBOX GATA element is occupied by tau1 and either GATA1 or GATA2, depending on the cell type. But it turns out that this precise sequence is shared by a number of other famous genes that are involved in hematopoiesis and erythropoiesis, including GATA2, ECLIF, KIT, RUNX1, and even some of the erythroid-specific genes as well. And so this conformation seems to be associated with many really, really important regulators of hematopoiesis and erythropoiesis. And one of the things we focus on in the lab is whether this sequence is shared or unique among different physiological processes in development and in regeneration and in cancer contexts. And can we identify specific features of cis-regulatory control that may contribute to regeneration following acute and chronic injury, which was kind of the focus of this talk, but also in context of embryonic development and leukemia progression as well. So here's what Yi Chao did. We were trying to kind of simplify the problem of gene expression and cis-regulatory control changes in the context of anemia. And so we chose the phenylhydrazine model for this experiment because it very rapidly induces acute anemia. Within really, I think, about 6 to 12 hours, these mice start to get anemic. About a day later, the hematocrit is below kind of what would be considered clinical anemia. This is also associated with an activation of erythroid progenitors within the spleen. That's kind of extramedullary erythropoiesis. And so we conducted an RNA-seq experiment on KIT-positive cells isolated from the spleen. And we did it in single cells so that we could identify the heterogeneity occurring in different subsets of cells within this context. And we did a few different analyses with this, and I won't go into all the details of this paper, but we identified certain subsets of cells that have unique activation of GATA1 and GATA2 activity. And so you can see here, I'm just showing you that in a control versus anemia context, we see increased activity of the GATA1 transcription factor within erythroid cluster 3 in this context. And these patterns seem to be a little different depending on which cluster you look at, which is super interesting. And as we kind of move forward, we're still digging into some of the interesting heterogeneity within these data sets and what it might mean in terms of important mechanisms that control the responses to acute anemia. But for Yichao's work, what he focused on was these EBOX GATA sequences. You can see that they're more frequently found at sites at chromatin sites that are associated with anemia upregulated genes. And that association is further enriched if you also filter for sites where you see occupancy of GATA and Tau1 transcription factors that bind to that enhancer. And the output of this analysis is a list of genes. And these genes are upregulated in the context of anemia. They have the same EBOX GATA cis element, and they have occupancy of the same transcription factors, suggesting a collective mechanism controlling the activation of these genes in the context of acute anemia. I've highlighted one here, SSX2IP, that I will come back to a little bit later. But that's what that was. The SSX2IP gene was one of the most highly upregulated genes following this analysis. And so I just kind of wanted to highlight for this group, some of this is very old information about the importance of the EBOX GATA element. So this was a cyclic amplification casting experiment. I don't know how many of you remember those old experiments, but this was a really nice validation that the EBOX GATA sequence is frequently associated with the formation of a larger complex that includes Tau1 and GATA, but it also includes the LDB1, which is a looping factor that brings the enhancer in proximity to promoter regions. Another project I won't have time to present today, but another project in the lab is really highly focused on the SAMD14 EBOX GATA enhancer, which is upregulated in the context of anemia and is required for survival in the context of acute anemia. So the next thing that we were interested in looking at in this data is the dynamic transcriptional states that are changing in response to anemia. And the interesting thing with what we discovered is that there's subsets of transcripts which are acutely activated and then repressed again very rapidly, right? So they come on early and then they get downregulated. There's another set of transcripts that are activated late. They do not come on until day 3, which is after the onset of anemia. And in some cases, these are transient. In some cases, they remain elevated at day 7, which is approximately the time point where the mice are recovered, right? The hematocrit has gone back to normal. We see other transcripts that are activated very late, really, I think after the mouse is recovered from acute anemia. And then finally, we see sites that are repressed. And just to kind of bring this back to the EBOX-GATA associated genes, they fit into the category of the genes that are turned on kind of in the middle, right? The intermediate activation state. So they're not active very early on. They're being activated kind of at the intermediate time. And then for the most part, many of these genes actually surprisingly remain elevated after the anemia has resolved. And so to further continue this project, we did an ATAC sequencing experiment, and we extended the time frame of our analysis from 7 days all the way out to 35 days. So this would be really a full month after the anemia has fully recovered. And so this ATAC-seq data gives us a more refined analysis of the early changes to chromatin states, but also the intermediate, and then finally these kind of what we call the deferred chromatin changes that eventually, for the most part, return to normal, although there are some notable exceptions that seem to be stubbornly changed even months after the anemia has recovered. And so this data set gives us the ability to answer some really, I think, important questions in the field. Can we predict which cis elements are controlling those anemia-activated genes? And what can we learn about the kinetics of stress erythropoiesis using the information of the signaling, or sorry, the transcriptional changes linked to the cis regulatory accessibility changes? And so in order to do that, we do an analysis called ATAC footprinting. If some of you may be familiar with that, but if not, it's a way to look at the holes in the ATAC data, and the holes indicate where there may be a transcription factor bound there. And the underlying motif that's beneath the hole in the ATAC data indicates that there may be a transcription factor bound to that. The important caveat is we don't know which transcription factor. We just know that that motif that typically binds those transcription factors is either more or less used within a particular context, all right? So what you're looking at here, if I can get my laser pointer, what you're looking at here is a control. So the PBS-treated mouse spleen compared to day one post-phenylhydrazine. And so when you see these stat motifs and AP1 motifs that are coming up on this side of this volcano plot, that means that these motifs are being used more by transcription factors after the onset of acute anemia. And we can look more granularly at some of that data and just showing you kind of an example of what that looks like. So here's the ATAC-seq data between day 0 and day 1. And then here's the footprint at those same time points, day 0 and day 1. So you can see at this KIF3B gene, there's a previously annotated cis regulatory element here, but you can see that its accessibility and its footprint score is increasing at that early time point post-anemia. Ooh, going the wrong way. Okay, so we wanted to know whether the AP1 footprint score was functionally relevant, right? So the experiment there was to take day 1 in phenylhydrazine injected mice, isolate their spleen, and then do an shRNA infection to knock down, in this case, the Jun transcription factor. We then cultured them for 3 days and did flow cytometry. And what you can see is if we knock down Jun, we get fewer KIT positive cells and more terminally differentiated TERMA109 positive cells, indicating that AP1 is promoting erythroid differentiation at early stages of, AP1 is actually inhibiting erythroid differentiation at early stages of recovery. And the way we interpret this is that it's involved in kind of the expansion phase of the erythroid progenitor cells, where they don't necessarily want to differentiate. They're kind of expanding. One of the questions I get a lot about this work is related to the use of phenylhydrazine as a model system. And so to address this, we have been collaborating with a variety of different labs to look at overlap between our acute anemia data and other chronic anemia data sets. And so this is work that was done by Theodosia Kalfa. They developed a Codanin 1 knockout mouse, and they crossed it to GATA1 Cree driver, and they did an ATAC-seq experiment as well, and RNA-seq experiments. And what we can see in comparing our acute anemia, and this is just the one time point, comparing our acute anemia activated cis elements to their Codanin 1 knockout mouse, we find about 7% of the genes are shared between those two data sets, suggesting there's some overlap there that may be relevant for chronic anemia contexts. And so all these changes we were seeing very early on in anemia, in the induction model of anemia, suggests that there's upstream signaling pathways that are involved in the regulation of transcriptional changes, but also the changes to the erythroid progenitors within the spleen. And so we wanted to identify what those signaling mechanisms might be and what those targets might be in the context of chromatin. And so I'm going to present, I think, two different ways that we've done that. The first way was a collaborative project with Len Zahn and Avik Chiduri. They had previously published a paper showing the chromatin signaling responses to BMP4 in human CD34 positive blood donors. And so we got our hands on this data set to ask whether these sites that were BMP4 responsive, and we know that BMP4 can control aspects of stress erythropoiesis and stress responses. So we wanted to know whether these BMP4 responsive cis elements in their data set overlapped with our data and may suggest that BMP4 may be a major driver of cis regulatory change and transcriptional change in acute anemia. This was further bolstered by their use of 18 different blood donors. And with this data, what they were able to do is actually look within the ATAC-seq sequencing data for variants, for actually polymorphisms that were different between one patient and or not a patient, a healthy donor and another healthy donor. And within that data, let's say you had one polymorphism where there was a link between the response to BMP4. So you'll have, in some cases, an ATAC-seq response that's higher if you have one version of the variant than another. So we were able to kind of look at selective responses that might be, where the variant might contribute to the response. And this is that list of associated genes with those cis elements. And you can see that many of them are activated in our acute anemia single-cell RNA-seq data set. Looking at this erythroid population 3, you can see these increased size of these bubbles indicates that more cells are expressing this set of genes in anemia context versus controls. And then I'm again showing you one example over here. Here's the ATAC footprint score in our mouse data set showing that there's an increased footprint at day 1 and day 3, and then a decrease back down to pretty much to baseline by day 7. If you correlate that with the Zahn lab human data sets at that same site at the CPD promoter, you can see that June is bound to that region. You can see it has H3K27 acetylation, and it has a BMP4 sensitive polymorphism at that site, suggesting that these polymorphisms are predictive potentially predictive of anemia responses. So the other way we're looking at this, and this was kind of initiated by a student in my lab, the other way we're looking at this is to try to identify common molecular profiles of signal response cis elements using multi-omics approaches. So this project was done by a graduate student in my lab who is bioinformatics focused. And so I don't know how many of you work with bioinformatics students, but they don't like wet lab. So we had to simplify this as much as possible. And I think that was actually a huge strength because it kind of allowed us to kind of almost in a reductionist approach, really kind of limit the variables. And so the major idea here is that he took well, I'll show you the experimental design in a minute, but the idea was to take RNA-seq data and ATAC-seq data downstream of signaling pathways, correlate it with promoter capture high Z and KIT-activated RNAs. So you've got the cis element linked to the KIT-activated RNA, or in this case, really any signaling pathway, and then to use all ENCODE ChIP-seq in hematopoietic data sets to try to correlate ChIP-seq profiles with ATAC-seq changes in response to signaling pathways and transcriptional changes. And then he fed all that through machine learning models to try to predict responses. All right, so the experimental design, he took QDEP2 cells, serum starved them, treated them with PBS or SCF, and did ATAC-seq and RNA-seq. So that's the whole experiment, right? So a lot of this, and I think it generated probably 10 years of work for the lab, just this one simple experiment. And so as you might expect, we see lots of cis-regulatory sites, potential cis-regulatory sites that are increasing or decreasing in accessibility. If we just do a simple motif analysis, we see AP1, which we had seen as our early day one footprint signature. And we also see this transcription factor EGR signature. We see the same two motifs when we do the footprinting analysis of the ATAC data, right? So we see a strong AP1 signature that's in the SCF treated side of the volcano plot, but not in the PBS treated side of the volcano plot. And then an example of that, here you see an ATAC peak that's coming on in response to SCF. The footprint score is increasing. And if you look at ChIP-seq data sets, it's occupied by EGR1 and June at the KLF17 gene. So when he fed these results through his machine learning algorithm, the output was both a ranking of cis-regulatory sites that would be predicted to be KIT responsive, and a list of transcription factors that are commonly associated with the KIT-activated activated cis-regulatory sequences. And so with both, we end up with a pretty powerful data set to do a lot of downstream analyses. If you kind of think about the accuracy of this system, if he takes the list of RNAs that are responsive to the KIT signaling pathway versus the ones that are not responsive, he's about 90% accurate. If you give him a gene, he can tell you whether it's KIT activated or not. And that's really just based on the transcription factor profiles that are found with those cis elements. To take that a little bit further, we developed a scoring system to rank the potential regulatory activity of each one. We call them KIT response elements, or KREs. So that's kind of where that language comes from. But based on transcription factor occupancy of the transcription factors that are involved, or sorry, that are predicted to occupy those sites. We don't yet know about regulation, but they're predicted to occupy those sites. Whether they are accessible on chromatin in the correct cell types. So in this case, since we're focused on the KIT pathway, the cell types we're looking for are the ones that are KIT high, like the CFUEs and the ProEs. And then we also integrated a few additional data sets into this analysis. We added, if you're H3K27 acetylation at that site, but also if you score, this is from Jesse Engritz's really interesting study using flowfish to identify active enhancers. But whether it's scored high on this ABC enhancer score list or not also gives it a higher score. If we then kind of put those scores on a map and look at evolutionary conservation, we can see that a lot of these really top scored KREs are highly evolutionarily conserved, which suggests that they're important and critical for development and or fitness. So the next experiment that I convinced this bioinformatics student to do was to try to perturb this network in some way to see whether some of our predictions turned out to be accurate. And so one of the genes that kept popping up on our list was EGR1, right? This is a gene where this is a motif where the footprint was increasing at that early time point, both in our in vivo acute anemia data set, but also in our HUDEP2 data set. This is also a mRNA that was strongly upregulated in the context of acute anemia and strongly upregulated when we treat the cells with SCF. And so the predicted cis-regulatory sequence controlling that response was kind of in the promoter region, about 1.2 kb upstream. So he designed a CRISPR interference construct that would prevent the upregulation of EGR1 in response to SCF. And you can see here that worked. So if you treat control cells with SCF, you see a 60-fold increase in EGR1 mRNA. But when the cells are expressing the CRISPR interference that targets this KIT response element, we don't see that upregulation. And we also see that as a consequence of downstream targets. This is a known target of EGR1 called spread2. And you can see that in that case, it's the CRISPR interference prevents the activation of spread2. So then he did the same analysis. He did RNA-seq and ATAC-seq to try to identify which of the the KIT response elements are EGR1 dependent versus which ones are independent, and can you build a genetic network to identify the features that are associated with sites that need EGR1 for activation versus sites that don't. And so I'm showing you a simplified version of that analysis where you can see in PBS treated cells, so these are control cells that have knocked down EGR1, you can see that the knockdown of EGR1 inhibits GATA occupancy or GATA cis element usage and promotes AP1 usage. When we treat cells with SCF, we see our control EGR1 signature is higher in the control versus the EGR1 knockdown, but we also see motifs suggesting that RUNX1 or some of the KLFs may be binding at higher frequency in the control cells versus the EGR1 knockdown cells. We can also do some validations of these cis elements. So these are 4 of the cis elements that we identified that control expression of associated genes that are KIT responsive. And in each of these cases, we see that when we target the response element with either CRISPR-I or CRISPR knockout, we downregulate expression of that gene, suggesting that this analysis and this prioritization approach is identifying functional cis-regulatory elements. And in some cases, the elements are pretty far away from the transcription start site of the associated gene. And so that analysis using the promoter capture high C, it really allowed us to kind of map that in finer detail. One last point I'll make about this is similar to the data with that we generated with the Zahn group, we can associate some of these cis-regulatory elements with polymorphisms that are linked to erythroid traits. And so this was a tool developed in Vijay Sankaran's lab, where he took the entire repertoire of polymorphisms that have been kind of previously just associated generally with erythroid phenotypes like red blood cell count, reticulocyte count, and really all the things that you can get from a CBC analysis. And we can see that there's an enrichment in our SCF treated conditions. There's an enrichment in ATAC-seq peaks that are associated with SNPs that are found in people with red blood cell count phenotypes. And here are just some examples of these SNPs. So we feel like this can be a really good tool to annotate these polymorphisms that previously had no ascribed functional link. It was really just an association to an erythroid trait. So to put that kind of all together into a model, we were sort of predicting these cis element responses using RNA-seq and ATAC-seq data linked to large published data sets to develop predictions. And then we can test those predictions using CRISPR-I or knockout approaches, and then kind of build these networks where when cells are expressing normal EGR1, we can identify circuits that are EGR1 dependent, that are regulating GATA occupancy and KLF occupancy. And then in EGR1 deficient HUDEP2 cells, that network gets disrupted, leading to changes in transcription factor occupancy on chromatin. Okay, so I will quickly run through the second half of this because I think it's also very exciting, and we're developing some really fun future directions. But what I showed you there was like just, I think, that early time point, right? Kind of day 0 to day 1, and the early activation, the BMP4 response, the SCF response. If we look at the later time points in this ATAC-seq data set, we see lots of unique changes and really a completely different transcriptional networks that are being activated. One of the starkest changes in the chromatin accessibility occurs between day 7 and day 14 in the post-recovery period. You can see that at day 14, there's a high usage of ETS motifs compared to day 7, whereas at day 7, we see a very high usage of GATA motifs. And this flip happens, again, between day 7 and day 14, which is past the point of anemia recovery, suggesting that the intrinsic transcriptional mechanisms of the cell involved in recovery is happening long after the hematocrit and red blood cell counts have already returned to normal. It also suggests that perhaps there is almost what we're kind of thinking about as like an overshoot or an overcorrection of some of the chromatin changes that are needed to restore homeostasis. And I think the best data that we have to illustrate that is if we look at, again, this is the tool developed in the Sankaran lab to link SNPs to link SNPs to red blood cell traits. And what you can see is that if we look at this over the full 35-day time point, we see an increase in the score of SNPs linked to red blood cell traits at these ATAC peaks, at these early time points. And then we see a sharp decrease below baseline, followed by a recovery to kind of similar to baseline. And we don't see that when we look at other blood traits, for example, white blood cell traits. There's really no pattern of change to the ATAC-seq peaks that we see at those time points, and megakaryocyte traits as well. So this dynamic response appears to be specific to the red blood cell lineage, again, because we're using phenylhydrazine to just ablate all the pure red blood cells. If we look all the way at day 35, and just go back one slide to just kind of highlight that, we see that for the most part, at these late time points, things return to normal, right? These cells go back kind of into their quiescent state, and they wait, I guess, for another anemia response, or another need for an anemia response. But at about 700 sites on chromatin, we see statistical changes in the accessibility after 35 days. And so this suggests that some of these sites may represent some form of epigenetic memory. I don't know if we want to call it that, but some memory of the anemia, the previous acute anemia event. And we're really excited about digging into whether or not these sites are associated with changes, durable changes to gene expression, and are linked to a functional benefit or deficit upon subsequent re-exposure to anemia stress. So to kind of put that together into a, do I have slides? Oh yeah, I have a couple slides after this. Okay, so to put that together into a model, if that's even possible. So here we have just kind of a schematic of kind of how the phenylhydrazine works on the hematocrit level, right? Mice get really anemic and they recover. This is associated with dynamic changes to transcription. Some of this might seem obvious, right? Like you induce acute anemia, of course, you get big changes to gene expression. But the genes that we're identifying within these data sets are really completely unstudied in many contexts. And so we think these might be kind of unique mechanisms that are involved in this response and may represent important therapeutic targets to target in both acute and in chronic anemia contexts. If we look at the later stages, we see a post-resolution switch from GATA back to ETS motifs, and we see that in a few cases, there may be durable responses that persist after the recovery period. Some of these anemia-activated genes have been associated with hematologic disease, including this SSX2IP protein, but the mechanistic relationships between the requirement for transcriptional activation and how cells use that for proliferation, survival, and recovery in anemia is really popular. Okay, so in the last minute, I'm going to our work with this P has chronic cis-regulatory element with an EBOX and a GATA. It's strongly anemia activated, but it's also strongly erythroid specific. So if we look at this data set from Laura Astiaso et al., we've used this data set a lot just to look at relative levels of gene expression throughout the hematopoietic system. We see that this SSX2 associated protein is highly specific to the erythroid lineage. Other things that have been published about SSX2IP suggest it's localized to centrosomes. And there's a few papers that have shown its association, its upregulation associated with increased leukemia cell proliferation. So in order to in the system of acute anemia and regenerative erythropoiesis, we developed CRISPR interference constructs to target the cis-regulatory enhancer, which is sort of shown here. And the controls for these experiments were kind of using this other region of the same intron targeted with a CRISPR-I. And so what we can show from using K562 cells is that the enhancer is functional, right? If we target the enhancer, we see downregulation of the gene. Here's another example showing that it's not just SSX2IP. There's another gene called TTLL12, which we're showing is also regulated by the same EBOX-GATA enhancer. We use an assay called FAIR, which is a chromatin accessibility assay. And using FAIR, we're able to show that not only is the gene expression down when we express the CRISPR interference construct, but also the accessibility of the enhancer is also decreased when we target. Functionally, I probably could have taken this top diagram off for this audience, but functionally, we use the shRNA to knock down SSX2IP and in stress erythroid progenitors isolated from spleen. And so we can efficiently knock it down. And what you can see from in the knockdown cells using CD71 and TUR19 surface markers, we see increased numbers of the mature differentiated erythroid cells. So not only does knockdown increase erythropoiesis, but also alters the cell cycle. This is a little bit of a dirty experiment because we did not synchronize cells in any way. This is just kind of asynchronous cells in culture, but using BRDU labeling and DAPI, we can identify G1S and G2 cells. And when we knock down SSX2IP, the relative percentage of cells that are in G1 increases quite a bit, significantly, and we see a drop off in the number of S phase cells in this culture. Also, if we do colony forming assays, we can see that both shRNAs against SSX2IP decrease CFU colonies by approximately half, suggesting that SSX2IP is promoting erythroid colony forming potential in stress progenitors. And so this is where we're at with the model. We kind of, again, we used our single cell RNA-seq acute anemia data set to identify SSX2IP and its enhancer as a potential response element that's controlling stress erythropoiesis. Under physiological contexts, SSX2IP regulation is controlling centrosome stability, and that's there are some really nice papers showing that if you lose SSX2IP, it destabilizes centrosomes, and you can get issues with M phase cell division. In the context of anemia, the SSX2IP expression is upregulated via an EBOX GATA element similar to SAMD14 or the GATA2 plus 9.5 site. And in these contexts, increased levels of SSX2IP is promoting progenitor activity and possibly promoting the progression of cells out of G1 and into S phase. When I was a graduate student, I heard a really great talk by Eric Lander when they had just sequenced the human genome. And he argued that they were publishing the parts list for the human genome, right? Here's the parts list, now put the airplane together. And I think he used airplanes as an analogy as well. I think the more we dig into kind of what these, not only just the coding regions of the genome, but the regulatory elements, is that the parts list is more problematic maybe than we thought it was going to be. A lot of these elements seem to have, it seems like a lot of these elements are unique in the context of physiological versus regenerative erythropoiesis, and we really don't know until we test it. The GATA2 9.5 element is a good example of that. It's required for embryonic development of the through its regulation of HSC emergence. The SAMD14 enhancer, which is nearly identical to that GATA2 element, is not required in that context. It has a completely different function in controlling anemia responses. And we're seeing similar things at other cis-regulatory elements genome-wide. But that's also very exciting. Testing function is essential to really understand what's going on. I think the more functionally validated enhancers that we have, the better our prediction models will be at downstream analysis of what other enhancers might do. But there's lots and lots of these that remain uncharacterized, and I think will keep us busy for a while trying to understand what's controlling which ones are controlling gene expression in the context of acute anemia and chronic anemia, but also how those cis elements are controlled upstream of that through the cell signaling pathways that are triggered in acute anemia. Okay, and I will end there, and I will thank the lab and the people that did this work. I already mentioned Rahul, and a current graduate student. He's about to leave the lab and is on the job market. Yi Chao left a few years ago. He kind of did that initial single cell RNA sequencing work. We have lots of collaborators. We were funded 3 or 4 years ago from the Hematology Center, the NIDDK Hematology Centers program, the Type B funding, which we used to develop the AP1 project. We've also been really lucky, I think, to have an R01 since 2021 that has funded most of this work as well. And I will be happy to take questions.
Great. Thank you so much, Kyle. All right. Questions for Kyle?
I'll ask a question.
Sharing.
Can you hear me okay?
Yes.
I can.
Yeah, great. So that was really wonderful. I think it's a great example of demonstrating the power of ATAC-seq and what you can get from those kind of data sets. I guess one question would be, as we sort of move into a post-genomics era where those kinds of technologies are just one part of the toolbox rather than an alpha and omega, there's this general sense in the field that transcriptional changes might only predict or encompass about 30% of a cell's physiology. So do you have any way of taking your data sets and getting a sense of, in terms of emergency erythropoiesis compared to non-coding RNAs and microRNAs and protein modification, degradation, localization, what fraction is being controlled by these gene regulatory networks? Is it most of it? Can you make any predictive sort of sense of that based on your data set?
Yeah, that's a great question. I mean, there's almost data. You can get to a point where there's almost too much data, and there's you get bogged down in kind of how the the protein level at the protein level, right? Whether that response is transcriptionally controlled or not. We are looking at a small window here of kind of the responses at the transcriptional level. But yes, layer upon that long non-coding RNAs and microRNAs and all the other layers of gene regulation that can lead to altered protein expression, and then whether that altered protein expression is even meaningful in the context of cellular behavior is a kind of another level that we haven't gotten to yet. I would love to do some single cell proteomics to try to put a number on that for this particular data set, because I think that would allow us to prioritize further. I do think that these the transcriptional responses are real, right? So they're real changes to gene expression. But whether it correlates with protein and how those feedbacks work that's a whole other layer we haven't gotten.
Has it been looked at, autophagy, mitophagy, those kinds of processes in this context?
So we have, that's a great question too. We have our project that I think is most mature in the lab on SAMD14, and in that work we have studied it in the context of autophagy, and we have a paper that just got out in iScience that has kind of linked the SAMD14 protein to control of autophagy in stress contexts, and So that's definitely, I think, the next stage for this, right? You have a gene list of transcriptional changes. Some of them probably are not correlated to protein level changes. Some of them might be correlated to protein level changes, but might not have relevant functions. So, we're trying to now take and, find naive graduate students to do the work and test it, is whether those genes have any function. I think that the cool thing is that if it doesn't, then there's another one on the list we can test, right? So that negative data then becomes kind of a project to look kind of at the next one, and try to understand the system a little better to where we can identify kind of vulnerabilities in targeting them, and to maybe I think our work with the polymorphisms and GWAS associations may allow us to build more predictive tools for understanding who might be at risk for developing acute and chronic anemia events, responses to chemotherapy, and things that may have important clinical value for treatment purposes.
Yeah, great. Thank you.
Yeah.
Other questions? No? Okay. Well, thank you so much, Kyle.
Awesome. Thank you, Kyle.
Thanks for inviting me.
Yeah, it was great to have you. Thank you so much for sharing your work. And I will wait for you to give me the go ahead on posting.
Okay. Can I just give it to you now, or do you need it?
Yes, you can give it to me now.
Yes, you have permission to post.
Awesome. Okay. Well, thank you so much.
Thanks, Kyle.
All right.
Bye.
Bye, everyone.
Bye.